Hello everyone. Today we'll be solving Cambridge International AS and A level biology paper 4 structured question February-March 2021 variant 42. Question number 1 A. The European eel Anguilla is a fish. The size of the eel population tend to remain relatively stable despite the eels producing large number of offspring. Such as two reasons why the population size of eels tend to remain relatively stable. So a population of an organism tends to, stay relatively stable because they are predated by other fish. The offspring compete for the same resources like food, and also if they increase in number too much, disease keep the population low. Question number 1, part B. Explain what is meant by the general theory of evolution. Theory of evolution states that organism changes over time. Organisms generally produce large number of offspring, and some of them have selective advantage for survival, so they get to survive for the next generation. And that's how they, slowly evolve in every generation. Question number C. The generation time of a species is the mean average time for one generation parents to the next generation of offspring. For example, the generation time of human is about 25 years. Figure 1.1 shows a graph of the relationship between the rate of evolution and the generation time for a wide range of different species. We can see generation time increases. All right. The generation time increases and rate of evolution with generation time decreases. If the generation time increases, then rate of evolution decreases as well. Describe and explain the relationship shown in figure 1.1. So in figure 1.1, we saw as the generation time, it increases, the rate of evolution decreases. Longer generation time means less reproduction. As a result, fewer mutation can be passed on to the next generation. So there is a less chance of evolution as the generation time increases. Part D. The Tautara Sphenodon punctatus is a reptile that is native to New Zealand. It is found nowhere else in the world. Figure 1.2 shows a Tautara. We can see in the figure 1.2 a Tautara. Tautaras have a slow growth rate and can live for over 100 years. Fossil evidence shows that there has been little morphological change in the Tautara over the last 200 million years. So this particular organism, it didn't change much in the last 200 years, 200 million years. This is a much lower rate of evolution than would be expected from the generation time of this species. Suggest and explain why Tautara have remained largely unchanged over the last 200 million years. All right. So, reptile-like organism, probably they are like, well adapted to their environment. So the selection pressure remained relatively reasonably constant. So as a result, there were fewer mutations. Mutations are extremes. So fewer mutations were selected for, and Tautara did not migrate to a different place. All right. So it stayed in the similar place. All right. It doesn't have the ability to migrate. So no separation of population from the original population, and thereby there was no chance for speciation to occur. Question number 2. The gray wolf Canis lupus is a large predator. During the 20th century, the gray wolf in the southwest Europe was hunted almost to extinction. Figure 2.1 shows a gray wolf. State the genus of the gray wolf. So the gray wolf is Canis, as we can see from here. Now the question says, Part 2, the question says, suggest and explain the effect on the biodiversity of the Southwest Europe if the grey wolf become extinct. You see, extinctions of grey wolf would decrease the biodiversity. Extinction of the gray wolf would decrease the biodiversity because gray wolf would be eating many more other herbivorous animals in that particular biodiversity. Herbivores may increase in number, which may lead to overgrazing, and there will be changes in ecosystem and food web over many years to come due to the extinction of the gray wolf. Part B. Gray wolves can have territories up to 950 kilometers square. Gray wolves can travel up to 1,000 kilometers to start a new population. State reasons why the mark release recapture method is not suitable for estimating the size of a gray wolf population. Basically, gray wolves, they're very big animals, so it will be difficult to capture them. And they can also be like dangerous to humans. Gray wolf has large territory, which is about 950 square kilometer. So they are unlikely to be recaptured second time. Gray wolf can migrate to other areas very easily because they can travel up to 1,000 kilometers to start a new population. Gray wolf may not mix with other wolf after they are being caught. So there is a small sample size. Mark release recapture technique, it can be inaccurate with gray wolf. Part C. In 1992, new laws were introduced across Southwest Europe to protect the gray wolf. Figure 2.2 shows the distribution of gray wolf population in Southwest Europe in 1970s and in 2012. No gray wolves from captive breeding populations were released into the wild in Southwest Europe during the period from 1970s to 2012. So we can see in number 1, the Iberian, number 2, the Sierra Morena, number 3, the Italian, and we can see in number 4, the Alpine. We can see the number 4 Alpine is a new population, which wasn't there in 1970s, and it's now present in 2012. Table 2.1 shows the sizes of the population of gray wolf shown in figure 2.2. We can see their increase in population. Sierra Morena, it decreased. In Italian, it increased. And in Alpine, a new population has started. Complete table 2.1 to show the percentage change in the size of the Iberian gray wolf population from 1970s to 2012. All right. Nearest whole number. We'll have to do this answer in the table in the nearest whole number. So we can see that the size of the population in 2012 is 2,500. And the original population we started in 1970s was 700. So minus 700. So percentage increase should be calculated with the original population into 100. So the answer is positive 257%. Part 2. With reference to figure 2.2 and table 2.1, describe the changes to the gray wolf population in Southwest Europe from 1970s to 2012. So, in the table, if we look at, we see that there is a overall increase, and we can see a new population. So there is an overall increase in gray wolf number. However, there is a decrease in gray wolf number in Sierra Morena, and gray wolf alpine population is a completely new population. Moving to question D, part 1. In regions of Southwest Europe where gray wolf populations are present, farmers are concerned for the safety of their livestock such as sheep. Suggest how governments can help farmers who are concerned for the safety of their livestock. Well, government can provide a compensation scheme for the loss of the livestock that the farmer might experience. Or D2, suggest measures that could help to protect wild population of gray wolves in the southwest Europe. So, because the question say suggest, so what kind of measures that we can take is we can establish a national park where, we can protect the gray wolves. And in the national park, hunting of that gray wolves is not allowed. Education and awareness programs can be set. Government can ban the hunting of the gray wolf totally, and the populations can be monitored so that we can find out that whether the population increases or not, or whether they're being poached or not. Question number 3. Vitamin A deficiency is a widespread public health problem. One source of vitamin A is carotene in the diet. Carotene can be converted to vitamin A in the body. White rice grains are not a dietary source of vitamin A because they do not contain carotene. Scientists have genetically modified a variety of rice to improve the diet of people who are vitamin A deficient. The grains of this genetically modified rice contain carotene. Describe how genetic engineering could be used to modify a variety of rice so that the grains contain carotene. So basically, genes involved in the production of carotene is first obtained from another species such as maize. The restriction endonuclease enzyme can be used, all right, for cutting the desired gene and the plasmid. The plasmid then can be used as a vector. DNA ligase is used for joining the plasmid and the desired gene together to make a recombinant DNA. The recombinant plasmid is then inserted into the rice embryos. The gene is then expressed within the rice embryos to produce enzymes to make carotene. In a 2018 report, the United Nations Children Fund, UNICEF, estimated that more than 140 million children are at high risk of vitamin A deficiency. 1.15 million child deaths are caused by vitamin A deficiency each year. Vitamin A deficiency is also a leading cause of childhood blindness. The recommended dietary allowance RDA of vitamin A for 5-year-old is 400 microgram per day. Genetically modified rice contains 12 micrograms of carotene per gram of rice. In 2018, 4 countries approved GM rice as safe to eat. The development of GM rice has been partly paid by the government. It has been agreed that seeds of GM rice will be made available to farmers in countries with high levels of poverty at same cost as seeds of normal rice. Some international organizations such as Greenpeace campaign against all GM crops. More than 100 respected scientists have asked Greenpeace to stop campaigning against GM rice. Discuss social and ethical arguments that suggest that supports the position of the scientist. So basically, if we do the math, 400 micrograms of vitamin A is needed by a 5-year-old child every day. So with a 12 microgram of carotene per gram of rice, this particular child, the dietary recommendation, the dietary allowance of vitamin A in one day can be easily met with 30 to 34 grams of genetically modified rice. All right, this can help prevent childhood blindness, and thus it will help prevent death of children. The seeds are affordable and scientifically proven to be safe. Greenpeace continuing to campaign would cause waste of government money. Question 4. Red algae are multicellular photosynthetic protoctists that contain phycoerythrin. Phycoerythrin is a photosynthetic pigment. Figure 1.1 shows the absorption spectrum of phycoerythrin, the action spectrum of red algae. All right. So we can see absorption spectrum of phycoerythrin and action spectrum of red algae. We can see phycoerythrin represented, absorption spectrum of phycoerythrin represented by dotted line. So we can see that it starts absorbing from 450 nanometer, and then the absorbance continues until 590 nanometer. Okay. However, the rate of photosynthesis, the action spectrum of the red algae can be seen that it starts from 440 nanometer, almost the same spot as that of the absorption spectrum of phycoerythrin. However, it stretches until 700 nanometer. With reference to figure 4.1, state the wavelength of peak absorption of phycoerythrin. So we can see the phycoerythrin, the peak occurs at this particular point. Okay, so we can see 550, 565, around 565 nanometer. Part 2. Explain how the data in figure 4.1 shows the phycoerythrin is not the only photosynthetic pigment in red algae. We can see that phycoerythrin does not absorb light above 590 nanometer. However, the action spectrum of red algae can be seen even after 590 nanometer. Photosynthesis still takes place. Part 3. Phycoerythrin is not the primary pigment reaction center for photosynthesis in red algae. Suggests the role of phycoerythrin in photosynthesis in red algae. So, since phycoerythrin is not the primary pigment, so it must be an accessory pigment. Phycoerythrin absorbs light up to, we can see 595 nanometer. It absorbs light up to 595 nanometer and passes it to the primary pigment, which then can be used for photosynthesis. Part B. Phycoerythrin is rarely found in plants that have green leaves. State the name of technique that can be used to separate and identify photosynthetic pigments. So to separate and identify photosynthetic pigments, we can use paper chromatography. Part 2. Explain how the results of this technique could be used to confirm that phycoerythrin is present in red algae and not present in plant with green leaves. So basically what we can do is we can perform the chromatography first, and then the RF value of the phycoerythrin, we can calculate it, or we can find it from a reference book. Then the green plant and red algae RF values of pigment can be compared together. The RF value of phycoerythrin will not be the same as the RF value of pigments in green leaves because they don't have it. Question 5. Many processes and actions in plants and animals are due to the movement of ions. Table 5.1 lists several ions, the direction of movement of each ion, and the action resulting from that movement. Complete table 5.1. Ion, sodium, direction of movement of ion from lumen of proximal convoluted tubule into proximal convoluted tubule cell, action resulting from the movement of the ion. So when sodium ions moves from the proximal convoluted tubule into proximal convoluted tubule cell, It co-transports glucose and amino acid. So co-transport glucose and amino acid. All right. We can find direction of movement into the guard cell, opening of stomata. So should be potassium ion. Potassium ion, when it moves into the guard cell, it causes water to enter the guard cell. And as a result, the stomata become turgid and it opens. All right. Movement of the ion into the presynaptic knob. Okay. Depolarization of presynaptic membrane. All right. So into the presynaptic knob, sodium ion enters. When sodium ion enters, depolarization occurs. Into the presynaptic knob causes the release of acetylcholine into synapse. All right. So it should be calcium ion. Calcium ion influx causes neurotransmitter to be released. Then next, hydrogen ion from intermembrane space into the matrix of mitochondrion. When hydrogen ion moves from the intermembrane space to matrix of mitochondrion, ATP synthesis can occur. If it passes through ATP synthase. Calcium ion from sarcoplasmic reticulum to cytoplasm of muscle fiber. So from sarcoplasmic reticulum to sarcoplasm to cytoplasm of muscle fiber, calcium ion will bind to the troponin. Ca2 plus ion binds to troponin. And as it binds to troponin, as a result, tropomyosin moves and sarcomere contracts. Part B. The venous flytrap is a plant that is able to capture and digest insect. It has modified leaves which have sensory hairs that respond to touch. When an insect comes into contact with the hairs, receptor potentials are generated. Name the ion that moves into the cells at the base of the sensory hair to generate receptor potential. So in venous flytrap, the ions that moves into the cell at the base of the sensory hair is calcium ion. Part 2. If 2 or more of these hairs are stimulated within a period of 20 to 35 seconds, action potentials are generated, causing the leaf to close quickly and trap insect. Suggest why it is beneficial for the plant for stimulation 2 or more hairs to be necessary before the leaf will close. Actually, this avoids wasting of energy. To avoid wasting energy. Part 3. The trapped insect is digested by enzymes released from leaf cells. Name the mechanism by which enzymes are released. Well, the mechanism by which enzymes are usually released in this situation is exocytosis. Part 4. Suggest why venous flytrap plants need to capture insects. Venous flytrap grows in soil that does not have enough nitrate, so they cannot make their own proteins, amino acids, and nucleotides.